Welcome everybody who are going to use BioLabDonkey program. In this video, I will demonstrate the functionality of Pdb Viewer. You need Pdb Viewer when you would like to open a Pdb file. In BioLabDonkey program, you can use a Pdb file containing data from X-ray analysis or from electron microscopy. Just press this button and then we can open this file. As you can see, this file contains only one protein chain, defined as protein chain A. If we select this line, then we immediately see the 3D structure of this protein chain. Here at the bottom, you can select which kind of mode you can use. Right now it's ball and stick. We can switch to wire, and also we can use space fill mode. It's quite small protein. Let's go back to the ball and stick. We can rotate, we can zoom in, zoom out. As you can see, at every amino acid residue position we have name, and these names are in yellow. If we select chain A in the table selected chains, then you will see the protein sequence. In this window, orange boxes represent alpha helices. We can see this color definition here. So in orange box is alpha helix and beta strand is green. In the case of 3D structure, we have atomic color representation here. For example, for carbon we have green color, for oxygen red, and so on. Here we have also list of non-canonical amino acid, which can be seen also in 3D model. On the right side, we see the table of other atoms and molecules which are present in this 3D structure. If we select, for example, this line, we will see sulfate. I guess it's here, this one. And again, we also can select other atoms and molecules. Let's select water. And water, as you can see here, is represented as red balls. In the ball and stick mode, we have quite overcrowded representation, and we can switch to wire. In this case, it's more clean view, and we clearly see the sulfate molecules in several positions, and also we see the water molecules. As you can probably already realize, in this case we have quite clear representation of alpha helices. For example, in this region we have alpha helix marked in orange in the same color as we see in the protein sequence, and also the green lines represent their beta strands. So the wire mode is more useful if you would like to see the alpha helices and beta strands in this 3D structure. To remove water molecules, again, we should select the corresponding line and then press this button, and then all water molecules will be removed. The same we can do with sulfate. Sometimes it's also useful to remove all names just to see the protein chain without amino acid names. If we would like to remove from certain region, we simply select this region. Let's say region here, And then we should check on this button and then press this button. By pressing this button, we will remove amino acid residue names. We can remove not only names, but also side chains and backbone chain. Let's say we would like to have in our 3D model only this part containing beta strands. To have this, what we should do, we simply select the upper region and then check on all 3 buttons and then press this button. Next, we select downstream region and again press this button. As you can see now, we have in our 3D model only this selected region. If we would like to analyze protein from this PDB structure, we can paste this protein sequence into the protein tab. We can do this simply by pressing this button. Now, as you can see, we have protein sequence from PDB structure. In this sequence, the X letters stand for missing amino acid residues. If we come back to Pdb Viewer, you can see that at the beginning we have histidine tag, and this tag is 21 amino acid residue upstream from protein itself. So we have histidine tag till position -16 and missing amino acid residue between position -16 and 0 position for alanine. And these missing amino residues are defined as red X here in this protein sequence. As I have said in the previous video about protein tab functionality, what we can do with this sequence, we can get secondary structures. By pressing this button, we will have these secondary structures. In this case, we have 2 possibilities. This is prediction, and we also can look at the real secondary structures by pressing this. And we see that we have more alpha helices in the case of real secondary structures than in the case of prediction. To improve the prediction, for example, we can, let's close this, we can add secondary structure motifs from this protein to the database, not default, but custom database. And next, when we will analyze proteins similar to this protein, with high probability, we will have much better prediction. And to do this, just to put the secondary structure motifs from this protein to our database, we should use this button. When we press this button, we have window where we can select database in which we would like to put these secondary structure motifs. Let's say this is mesophilic organism, and we check both protein location, and then by pressing this button we will add secondary structure motifs to the custom database. Now let's come back to the protein tab. When we press this button, we have option for mesophilic database, and we know that we added secondary structure motifs from this protein to custom mesophilic database. Let's do this, and again check also to both options, cytoplasm and membrane, and then we can press this button, and now as you can see we have the same secondary structure motifs for prediction and for PDB. Additionally to the beta strands and alpha helixes, in the case of prediction, we have also regions defined as turns. Let's close these windows and come back to the Pdb Viewer. We have also option to put the protein sequence into the alignment tab. If we press this button, you see the protein sequence in the alignment tab. Here the name of file and protein sequence. Let's come back to the Pdb Viewer. At this moment I would like to finish this video, and in the next video I will talk about RNA differential analysis. So this will be this tab in the program, and as you can see we have here two options: one, differential analysis for mRNA, and also for microRNA at the bottom of this window.